Good morning, students. Last class we have discussed about the blood and that is formed elements such as the plasma in the blood fluid and circulation. So again we have to just reminding that one again one more time. So blood consists of the fluid matrix, that is plasma, and the fluid matrix, that is plasma, and there is the formed element. You can see that one here. The red blood cells, white blood cells are present here, and platelets are present. These are called the formed element, and this is called blood vessels, and vessels walls are also there. So here three layers are present for the blood vessels, that we'll discuss after. Next one is coming plasma. Already have discussed that one. So here the formed elements also we have discussed: erythrocytes, leukocytes and platelets. And erythrocytes, what is their amount, what is their lifespan, where they are originated, what is their gravure, all these things we have discussed last class. Here leukocytes also two types: granulocyte, agranulocyte, and neutrophil, eosinophil, basophil, as well as the monocyte and lymphocyte. All these things we have discussed last class. And platelets also, their amount, that is amount is coming here 1,50,000 to 3,50,000 here, and per millimeter cube of blood. And they are actually cell megakaryocytes they are actually carrying. So these are coming here about the platelets here. Next we are coming, these are the different formed elements. One is here it is coming platelets here, red blood cells, neutrophils. It is neutrophils actually. This is then monocyte, lymphocyte, eosinophil, basophil. All these things already we have discussed. Now blood groups also we have discussed here. A, B, O, four types of blood groups are there: A, B, AB and O, and Rh positive and Rh negative. Here ABO grouping. So you can see here very fine diagram is also given here. So look at here, it is A blood group. If it is A blood group, so it is coming it is actually named on the basis of what is the antigen present on the we can say the RBC. Who are the antigen present here? So definitely in case of the A blood group, antigen A is present here. So in the actually plasma, what is the antibody present here? Anti-B antibody. Anti-B means actually if it is anti-A is coming, so it will be clotting. Both will be clotting here. So that's why in the plasma, in case of A blood group, anti-B antibody is present. In case of B blood group, anti-A antibody is present. In case of antigen A and B, both are present there, so no antibody must be present here, otherwise clotting will occur. And then neither A or B, we can see that one antigens are present here, so both the antibodies can be present here. And O is known as the universal donor because it can donate the blood to every blood group there, AB, ABO. Every blood group can take O blood group, but AB is also called as universal acceptor, so AB can take blood from any other blood group, but type A actually can take blood from A or O. Type B, once B blood group can take B and O, AB can take from we can say all the other blood group, but it can give only AB blood group. Okay, this is the ABO grouping is coming. Next one we are coming for Rh grouping. So today we are discussing about the Rh grouping here. So Rh actually named on the basis of the Rh antigen. Rh antigen is similar to one present in which monkey? Rhesus monkey. That's why it is known as the Rh antigen. It is also observed on the surface of RBC of majority humans. Such individuals are called the Rh positive or Rh plus B. A person without Rh antigen is known as the Rh negative. Now significance, what is that? An Rh negative person, if exposed to Rh positive blood, will form the specific antibody. Suppose if Rh negative antibody or Rh negative person, if exposed to Rh positive blood, so what will be happening there in their blood or in their body? The specific antibodies against the Rh antigen will be produced. Hence Rh group should also be matched before the transfusion. So this is the, here you can see the blood type, that is RBC with Rh antigen, and here it is coming RBC with no Rh antigen. You can see that one antigens are not here. These are the yellow color, these are antigens actually. These antigens are not present here. Okay, next one we are coming, the coagulation of blood. What is the meaning of coagulation of blood? So blood coagulates in response to any injury. So when any injury is there, any cut is there, blood is flowing from that injured area, and then after some time all of we can see the blood is clotted there. So what is actually, that is called coagulation of blood, and what is the mechanism of the coagulation of blood there? So coagulation is a mechanism to prevent the excessive loss of blood in case of injury. Clot is a dark reddish brown scum which is formed at the site of injury over a period of time. After some times what you can see, and clotting occur, dark reddish brown scum occur there. So that is called the clotting. This is formed by a network of threads called the fibrins, in which dead and damaged formed elements of blood are trapped. So we can see now how the mechanism is going on. So look at here, process of blood clotting. Here you can see the inactivate fibrinogen is present in the plasma. It is converted by the enzyme thrombin into active fibrin. So first one is the fibrinogen which is present in plasma, that is converted into which one? Fibrin. And thrombin is formed from the inactivated, or we can say inactive prothrombin. So prothrombin is converted into thrombin, and fibrinogen is converted by the enzyme prothrombin into active fibrin. So now it is coming, an enzyme complex thrombokinase is responsible for this conversion. The complex is formed by a series of linked enzyme reactions, and the process involves a number of factors present in the plasma. So now we are coming here the point. So look at here. So first, what is coming first? Injure the tissues and platelets, they release the clotting factor. What is the clotting factor? Prothrombin, activator and calcium ion. Both the clotting factors are released by injured tissues. Then prothrombin activate towards the product, which one is? Prothrombin is converted into thrombin. So prothrombin convert हो गया किसमें? Thrombin में. Then fibrinogen is prothrombin converts the fibrinogen into fibrin. Then fibrin, what they are doing? They form a mesh of trapping the red blood cells and the platelets there, and the bleeding stops. Okay, and the bleeding stops there, and the clot hardens, and the, we can say, become a smaller, and new cells grow on the repair wound cells there. So first, what is the factor here? These are the after the clotting occurring. So prothrombin activator and calcium ion. So prothrombin prothrombin activator converts the blood prothrombin to thrombin, and thrombin converts the fibrinogen to fibrin, and fibrin is adding with the erythrocyte, that means blood cell, to convert it into, we can say, the blood clotting. So this is the actually bleeding stops there. They are forming a mesh like structure, and the bleeding stop there. So that is our main mechanism here. Mechanism is the prothrombin converted to thrombin. Thrombin plus fibrinogen is fibrin. Fibrin plus erythrocyte equals to blood clotting. So this is the main process of blood clotting here. Next one, we are coming the blood vascular system. What is the blood vascular system here? Means blood vascular system here, it is actually given by demonstrated by William Harvey, and they have actually, where higher animals have the well developed circulatory system to transport the substance in the body, can be done very effectively. In them, the circulatory system consists of central pumping organ, that is called heart, and the various blood vessels. So what are the main component here? Heart as well as the various blood vessels. Who are the blood vessels here? Arteries, veins and capillaries. Arteries conduct the blood from the heart to other tissue. Veins bring the blood from other tissue to the heart. Some of the invertebrates and all vertebrates possess this system, and this circulatory system is actually discovered and demonstrated by William Harvey. So here it is consisting of heart as well as the blood vessels. Blood vessels consisting of which one? Arteries, veins and capillaries. Arteries conduct the blood from the heart to other tissues, and veins conduct the blood from the other tissue to heart. So here two types of circulatory systems are there. One is open circular system, where the we can say in many advanced invertebrates such as prawn, insect, arthropoda में हम देख सकते हैं कि the blood does not remain confined to the blood vessels but it flows freely through the body cavity and channels called the lacunae and sinuses. Means cavity is known as the lacunae and channels are known as the sinuses. Means blood is not carried out by any type of blood vessels there. Blood is circulating through the means heart and the tissues. So that is why the body cavity is known as the hemocoel. The body cavity which is filled up by the blood is called hemocoel. It is given here, and the blood is hemolymph. So body cavity which is filled up by the blood is known as the hemocoel and the blood is hemolymph. In insect, the tissues and are in direct contact with the blood. Hemolymph circulates and the whole body due to the contractile activity of the heart. So this is called open circular system. There the blood is flowing, not flowing through the vessels there, and they are actually flows through the freely, that is body cavity and body channels, that is lacunae and sinuses and sinuses, and the body cavity is known as hemocoel and the blood is called the hemolymph. That is called open circular system. Means as such the arteries, veins and capillaries are not present here. Next one we are coming for the closed circular system. In the closed circular system, you can see in closed circular system the blood flows through proper blood vessels, जैसे arteries, veins and capillaries, and arteries within the tissue divide into arterioles which then branch further to form the capillaries. तो पहले artery divide हो जाते branching into arterioles, arterioles again branching into capillaries, capillaries again then unite to form the venules and which come out in the tissues and veins. So this is actually condition. So first the arteries branching into arterioles, arterioles into capillaries, again capillaries into venules, venules into veins. So this is actually the venules again combine to form the vein. So that is the condition. Capillaries again combine to form the venules. So in that way it will be going on. Arteries have thick, elastic and muscular walls which are made up of three contractile layers, concentric layers. What is that? Tunica externa, tunica media and tunica interna. That is already it is easy to remember. Externa, media and interna, it is the layers of the vessels, that is arteries. And all these layers have got smooth or involuntary muscle. Capillaries are extremely fine, thin blood vessels with the walls which are made up of single layer of endothelial cells. So capillaries are very fine. So whatever the exchange of nutrition and gaseous exchange occurring, that is through the capillaries only, because it is very unit cell membrane structure, means very fine and delicate it is. The muscles and elastic fibers are absent in them. These capillaries are highly permeable to water and small macromolecules. Various nutrients, respiratory gases, metabolites and other substances are exchanged between the blood and tissues through these capillaries. So capillaries are actually main part through which the exchange of the respiratory gases as well as the nutrients are occurring, because they are very fine, they are very delicate and they are unit membrane structure. Whereas arteries, in case of arteries they have the thick elastic muscular walls. In case of capillaries no muscles are present there, only they are responsible for the we can say the exchange of gases. We can say the arteries, they are actually branching into arterioles, arterioles are branching into capillaries, again capillaries are combined together to form the venules, venules are combined together to veins. So in that way the circulation is going on, that is called the closed circular system. Next one we are coming, what is the difference between the open and closed circular system here. In open circular system the blood flows through large open spaces and channels called the lacunae and sinuses among the tissues. Means lacunae and sinuses means it is coming about that cavity and the channels here. And in closed circular system blood flows through the, we can say the closed system here, through the closed system, that is and the chambers called the heart and blood vessels. टिश्यूज इन डायरेक्ट कॉन्टैक्ट विद द ब्लड, क्योंकि कोई ब्लड वेसल्स नहीं है तो टिश्यूज कॉन्टैक्ट होता है, डायरेक्ट कॉन्टैक्ट में रहता है ब्लड की, but blood does not come in contact with the tissues. Blood flows very slow, blood flows very rapid here with a high pressure, क्योंकि इसमें क्या वेसल्स रहता है तो डेफिनेटली हाई प्रेशर से क्रिएट होता है वेसल्स के थ्रू, आर्टरीज एंड वेन्स के आर्टरीज के थ्रू. Exchange of gases nutrient takes place directly and exchange of nutrients and gases pass through the capillary wall. Okay. Next one we are coming for the, you can see here, less efficient as volume of the blood flowing through a tissue cannot be controlled as blood flows out in open space. More efficient as the volume of blood can be regulated by contraction and relaxation of the smooth muscles of the blood vessels. It is more efficient. Open circular system found in higher invertebrates like most arthropods such as prawn, insect, etc. and closed circular system found in where echinoderma, some molluscs, annelids and all vertebrates. तो आर्थ्रोपोड्स में ओपन सर्कुलर सिस्टम फाउंड होता है, बाकी इधर इकानोडर्मास में या मोलस्का या एनिलिड्स and all vertebrates में which type of circular system we can see? Closed circular system. Respiratory pigment present in the is dissolved in plasma. RBC है ही नहीं इधर, तो definitely plasma में present होता है. Respiratory pigment is present in maybe it is present, maybe it is in a dissolved form of plasma, but actually it is held in RBC. ये RBC में रहेगा, ये plasma में रहेगा. Plasma मतलब क्या है कि content of the, we can say the matrix of the blood, it is called the plasma. Okay, so arteries and vein, this is the difference here. Arteries is the blood flows away from the heart. Here blood flows towards the heart. Blood flows with jerks and with great pressure because arteries have the muscular walls. Blood flows here smoothly and with less pressure here. And they always carry the oxygenated blood except the pulmonary artery, and they always carry the deoxygenated blood except the pulmonary vein. Lumen of arteries is small. Lumen means a cavity. Lumen is the vein is large, and valves are absent क्योंकि इसमें तो we don't have, they are carrying the oxygen rich blood, so they don't have to stop the backflow of the blood here. But here the semilunar valves are present to prevent the backflow of the blood. They are deep seated, means they are actually present, not present superficially. Here they are present superficially. ऊपर ऊपर रहता है. Their wall are elastic, thick and muscular. Their walls are non-elastic, thin and collapsible. This is not collapsible, and they are collapsible. So these are the difference between the arteries and the veins. Okay, next one, next is the heart. So next we will discuss about the heart here. So before that, we have to discuss one thing here, that is regarding the RH group. So all of you know where the RH antigen is present, they are called the RH positive, and where it is absent, that is called the RH negative. Now one special case of RH, here it is coming about the incompatibility. What is the special case here? Okay. So what is the special case here? That is the special case of RH incompatibility, that is mismatching is observed between the RH negative blood of a pregnant mother with the RH negative blood, means RH positive blood of the fetus. Suppose if the mother is with RH negative, okay, and we can say the baby is with RH positive. So at that condition, okay, what you can see here now. So it is coming. So RH positive blood of the fetus. RH antigen of the fetus do not get exposed to the RH negative blood of the mother in the first pregnancy, as the two bloods are separated by the placenta. However, during the delivery of the first, we can say there is a possibility, first child, there is a possibility of exposure of the maternal blood to the small parts preparing antibodies. So